Welcome back, General Ecology. Talking about parasitism and infectious disease today, which is essentially chapter 15. There are a lot of good stories. I told you of quite a few that are sort of in my back pocket. In your book, there's a lot of good ones. You'll see some overlap between those stories. But look in the book, look at those examples, and you don't need to get the case study specifically memorized necessarily for my classes, but it does help to sort of get what's the point of all these stories? What's the larger picture? What is he trying, or is the book trying to tell me? And of course, the book does a very good job of laying out what's the context and why this matters. So make sure you do that. Don't just kind of sit there and watch like it's a Discovery Channel storyline of weird things that infect insects and pop out of their foreheads. Get the ecology. Think about the ecology. All right, so this is part 2, and I'm going to get into some human examples in a minute. But here, parasitoids, again, I think I mentioned in the last segment that sometimes you could see moth caterpillars hanging from a thread, and their insect parasitoid will come in and drop an egg inside. And that could be, well, that's the end of that particular caterpillar. Its fitness is about to get converted into wasp fitness. And this happens a lot. It is a whole topic of research. And Dylan Perry in our department does quite a bit of work on parasitoids. So if you're interested in these kinds of things, you should probably talk to insects. So Dr. Weir is working with the fungi. I do a little bit. Dr. Perry and other entomologists would be aware of these parasitoids on insects. And I love this story, this next story here. This kind of blew up in a sense while I was getting my PhD, and someone came and gave a departmental seminar while I was, second or third year PhD, and I just was thoroughly fascinated by this. I don't know why I love these stories so much. I guess it says something about me. But this is a tobacco hornworm. They are common. They're great big green things. You could buy them at the pet store to feed your larger critters. They're voracious. They eat a lot, so you can see the leaves. They could be on tomato, tobacco. Those are related species. And I don't know if there's different species of hornworm on the different plant species, but you get the idea. They all look kind of greenish. What happened here? What the heck happened here? What's going on there? Let me just say that ain't normal, okay? That ain't normal. So what happens is the parasitoid wasp comes in and lays her egg on the unsuspecting worm who's eating away and happy, or lays a bunch of eggs, right? Drops them in there. At the same time, she's depositing a little bit of fluid, and in that fluid is a virus. The virus causes gigantism in the worm. So when infected, those worms get humongous compared to the other ones, twice the size, big, fat, plumpy things. And as the larvae of the wasp develop, there's more food available to them because of the action of the virus. So the virus is getting in there and taking over the metabolism and whatever the machinery of the worm, causing it to get big, which is good for the virus, but it also is good for that wasp. So the parasitoid wasp lays her eggs. I'm saying tobacco hornworm. I don't know if it would work on the potato or tomato, but probably. With those eggs, she's dropping in some viruses. That causes gigantism in the caterpillar, and it's kind of like Little Red Riding Hood, the more food to feed her babies. And that it says left panel. Obviously, I meant right panel. The right panel shows the larvae exiting the enlarged host caterpillar. Those white things are the larvae exiting, and they're going to become adults. So, all right, let's start looking at humans. And one of the things about some of these pathogens, and where humans, well, not just humans, but these pathogens or parasites, they have really complicated life cycles. So the picture on the left, you've probably seen something like this in your biology class, or if you've had botany. That is the wheat rust, okay? And it also can occur on white pine blister rust, which is not in that life cycle is the wheat rust, but this also happens with white pine. And that white pine will not complete its life cycle without ribes around. So it's caught multiple, as some of you may know, the different rusts will have 3 or 4 or even 5 different spore stages, and they have different hosts. So they somehow override resistance of 2 different hosts in order to complete their life cycle. And with the wheatgrass one, I talked about that earlier in an earlier lecture where the federal government gives you which cultivar of wheat you grow in order to have resistance against the wheat rust. It's everywhere. This stage, or these stages here, the asexual stages, crank out so many spores that they readily infect across the board, and you have to be very careful. They're always present. This is where outcrossing occurs, and through this stage up into the basidiospore production. And so that one's a little less ubiquitous, but once it gets onto the barberry and then gets onto the wheat, then you've got widespread contamination of everywhere. And it's a pretty smart strategy. The funny thing is that humans were not so smart, because when they saw that there was 2 different hosts, well, they couldn't get rid of this host because that's the crop. So they said, oh, let's eradicate this host, the barberry. And so they had this big program where they said, go out and pick all the barberry in your area and get rid of it. Impossible. You can't get all of it. And it only took a few hiding out there to produce enough to get onto the wheat, and off we go with the next infection. Anyway, 2 hosts back and forth on an annual basis. And we have the same thing in a number of cases. This one is showing you malaria. This poor person has been infected. The mosquito takes a blood meal, but also deposits the organism and causes some pretty dramatic issues for this poor person. But again, it's going from mosquito to human host back and forth. And that's, to me, that's intriguing because, again, I'm very interested in the genetics of resistance and virulence and how the organisms evolve and play that dance. Very successful at it. And someday, I think malaria will be solved if we can tag one or the other phase of this resistance interaction and give humans a better ability, or perhaps the mosquitoes, I don't know. You can't get rid of the mosquitoes, that's not going to happen. They used to spray walls in Florida with DDT, thinking they could get rid of mosquitoes that way. What they did is selected for mosquitoes that didn't land on walls. So mosquitoes just went ahead and did their thing and continued to infect humans. Okay, here's another one. You're very familiar with Lyme's disease, I'm sure, caused by Borrelia burgdorferi, transmitted by ticks to humans, but it is transferred, vectored in the environment by deer, by birds, by mice, and it's when it gets onto the human that the disease is carried over to us. One of the things about this, and I think I mentioned this in an earlier lecture, if there's a bumper crop of acorns or butternut or something like that in our area, you can kind of expect a bumper crop of mice. If there's a bumper crop of mice, you can kind of expect a bumper crop of ticks. Bumper crop of ticks, look out for Lyme's disease. I've had it. My daughter had it. I remember going into the doctor saying, well, I think she's got Lyme's, and he says, oh, probable or potential Lyme's. He said, how do you know? And I said, well, she's got these big migrating red rashes on her leg, 3 or 4 of them. He writes down his notes. Thinks she has migrating red erythema, or whatever you say, rashes. So then he looks over at her leg, and he's like, oh, she's got Lyme's. I'm like, yes, she did. Yes, she did. She does. And we got her cured. She's fine. Anyway, be very careful when you get hit by this. It's a bacterium, the Lyme is, and it's easy to cure if you get it early, just with antibiotics, doxycycline. So if you get any sort of feeling of fever and chills and just feeling kind of crummy, and you've had tick bites, or we're in an area where ticks are, because sometimes the tick will bite and drop and you don't see it, right? Sometimes it attaches and you pull it off. But just be very aware and go to the doctor and say, there's a possibility I got Lyme's disease here and I'm feeling pretty crummy. Here are my symptoms. So easy to solve. If you don't, it goes into a nasty phase 2 and phase 3 where you can have some real trouble, all right? Hard to get rid of because it goes into nervous tissue and heart tissue. Okay. The other thing to note is that it's not just Lyme disease being carried by ticks anymore. Again, talking to Brian Ledette, Dr. Ledette, you will learn about a lot of different microorganisms that are being transferred by ticks. And it's a pretty scary world now because the ticks are becoming incredibly abundant out in our area, probably with climate change. Okay, a couple of lessons we can learn from a lot of these storylines. So here are deaths on the right side and infections on the left side of West Nile virus. I forget where this was, but it could be in the US somewhere. It's in your book, so I'm sure you can find it. From 2000 up to 2015. The main thing I want to point out here is actually how to read a graph. Okay, we know West Nile virus is in our area, or in North America now. And if you look at the purple line, this is the left axis, y-axis. So the numbers go up to, well, up to 10,000. The purple line is going up to about, let's see, this is going up to about a little over 4,000. Those are the infections, okay? Look at the deaths, going way up, 10,000 in 2003. Usually they're more closely related. This is a really nasty virus. Most people that get it might actually succumb and die. Certainly those who are elderly and have immune-compromised systems can't kick this thing once they get it. It really knocks them out. So we're learning here, I'm using this to sort of lean into some ideas about COVID-19. I do want you to see that the two axes are different. This green line is not 10,000 deaths, it is 600 deaths. So although they are correlated, infections to deaths, the green line is on this axis. I'm sorry, I botched that. Make sure you get that. Deaths are far fewer than the infections. So that's one thing. The two axes are different. The infections are not the same as the number of deaths, but they are highly correlated. There is a very strong impact once you get infected. And I guess I'm using this to say in today's world with COVID, there's a lot of discussion about whether we should be or not be testing so much and what's the point of testing and does it even matter? Heck yeah, it matters. If you know how many infections are out there, you can make a pretty good prediction of how many deaths are going to be there, okay? It's going to be fewer, but it's highly correlated. And why ignore the data? So I think it's an amazing factor that we are often sort of listening to people who are not scientists, are not medical researchers, and they're telling us not to worry about COVID. Now I'm talking to you today in July, and I think finally the politicians, certain politicians, are finally getting it. But man, can we use some science in our public discourse, all right? So a couple more cases here to discuss. Here we have, I wanted to highlight, there's one thing. I talked a couple segments ago about horizontal resistance and vertical resistance. The book brings up a similar sort of use of terms, in this case, horizontal transmission and vertical transmission. Very different. Vertical transmission is parent to offspring, all right? And so they're showing an example of this great blue heron transferring probably just through her blood, getting into the female, into the egg and into this chick, a disease such as chlamydia, all right? That's vertical transmission, parent to offspring, all right? Horizontal transmission would be the mosquito hits this bird and then carries it to another bird, okay, and brings with that West Nile virus, or to a human, okay? Another horizontal transmission may be, so that would be within the same species, but you can also have them crossing species, for instance, to humans. Or they're mentioning here bird flu, going from birds to humans also. Here's horizontal transmission with multiple hosts with helminth, little roundworms, going from snails to frogs to birds, a very complicated life cycle. So transmission, vertical and horizontal, know those. And then there's resistance, vertical and horizontal, know those. So just to reiterate then here, vertical is parent to offspring, horizontal is directly between compatible individuals or indirectly through a vector, and it could be within or across boundaries, but it's not parent to offspring. And then there's another concept that's not in that particular figure, but reservoir species. So I think the best example might be the American chestnut. The chestnut blight can occur on other tree hosts in our area. It can occur on oak and Chrysolepis. I only know the genus name, sorry. Tan oak? No. Anyway, and it's always out there and just kind of in very low numbers. So the chestnut themselves will get knocked right back to the ground, so to speak. But these other hosts are reservoir species, so the chestnut might disappear, the blight is largely gone, but then those reservoir species, it's just sort of hanging out in low numbers, not killing the trees, and then you plant chestnut, if it's not resistant, and eventually gets hit. So you have to be careful with these reservoir species. They don't get hit hard, but they keep the organism around. And finally then, with the lessons that we learn, here's something called the SIR model for susceptible individuals. Over here on the left, initially will be very high and they'll get knocked down. And they'll either move over to recovered individuals, this green line, or just infected and maybe even dying individuals or dead individuals. So this SIR, S, susceptible, I, infected, R, recovered. We can use these models in every infectious disease out there and get a handle on how to control it. We've been hearing a lot about flattening the curve of infected individuals with the COVID this summer. Bring this down. Well, okay, but if you're just bringing it down and preventing infections, you might still have a lot of susceptible individuals out. So the more interesting thing is to create a vaccine that actually basically mimics recovery and gets these individuals over to the green line, all right? So this is the SIR model. You can read about it. If you could do a Google search on it and COVID, and you will find information out there. It stands for I, S, and R is individual, susceptible, and recovery. And it is a simple model like all the others, but we can use it. As a simple model, as I say here, it doesn't assume or allow for new susceptible individuals to be born, right? They're either susceptible and then they move to one of these others. You don't have a reintroduction of susceptible individuals, which is not real. That's not reality. But the modeling can be used effectively to control the disease a little bit better. So a little bit of science goes a long way in public discourse and our ability to react to these infectious diseases. And as a final point, here is a bunch of data on measles out of London. And this is out of your textbook. So very cyclic, year to year. There were peaks and valleys, high infections, low infections. And what was going on up until the vaccine was produced is that individuals were getting infected. Most of the susceptible individuals were dying. Those that recovered then reproduced new resistant individuals for quite a while. But in the absence of the pressure of having measles around, because it was no longer a disease of the area, susceptible individuals started to get born, more and more and more, and then pretty soon there were a lot of susceptible individuals, and the measles outbreak takes off, and it repeats the cycle over and over and over. And then in the 60s, early 70s, a vaccine was developed. The first thing you notice, the peaks are attenuated. Not as many people get the measles because of the vaccine. The other thing is, through repeated usage, the measles starts to go down completely because it's just not working out for the measles. They cannot maintain a presence. And so in the 90s, we actually almost got rid of the measles. I believe there's been a couple of outbreaks in a couple of years now, but these are in part because of the no-vaxxers, people that won't take the vaccine, which is basically building up the susceptible individual pool again, whether that's in the US or in other countries like Iran and Iraq, where they don't trust the medical professionals who say you should take this vaccine because they don't know what's in there, and a little bit of misunderstanding goes a long way to causing some serious trouble down the road. So anyway, let's see, what did I say here? These are the outbreaks in London. Note the cyclic nature. I did give you the answer to that. What's the cause of the pattern? You can read about it also, or you can think about it right now. You probably could remember. Note the 90s. In the 90s, we got a vaccine. Hopefully today we're talking in the same way, and I'll be lecturing or having a chat group with y'all about COVID and its vaccine that is now working for us. And although the disease, you can get that vaccine, it will still be problematic for decades. It'll still hang around, reservoir populations of people that aren't using vaccines. And so we definitely have to still manage it, but at least it's a lot easier to manage this measles than it used to be. And I think the COVID will be the same way. We will be dealing with COVID for a long time, there's no doubt. But we can manage it now with proper vaccines, and probably more than one by the time we get to this time in the fall when we're talking about this lecture. Oh, right. Now, what's up with these elk? Moving on. It's another thing that infects us. Those of you that are hunters know this story, at least to some degree. I'm going to give you some of the ecology and biology around it today. Those elk, if you look at the left-hand panel, it's drooling, all right? And it's also pretty skinny. Ears are floppy. It's not feeling very good. The next one over, you get more sense that it's really skinny, emaciated. Again, the ears down. This is not a happy animal. And then the far right panel, the same thing. So what do these animals have? Chronic wasting disease. So it is a disease. It is transferred through urine to different animals, across species boundaries to some degree, but we don't know how much still. You could take brain tissue, that's a big hint about what we're dealing with here, out of one of these sick animals and inject it into another animal, and it will get sick. You can get urine, and these animals tend to go lick each other's body parts and pick it up through the urine. This is how it's spread. This is a disease of the brain. Encephala is brain or head. Pathy is pathological, so it's a brain disease. And spongiform refers to the spongy nature of the brain tissue. It's holy once the disease sets in. So when I first started lecturing in 2001, this was a huge story, CWD. It's less so now. We're a little bit more aware of it and a little less concerned, but we shouldn't be. It's a very important situation. Before the infection, you could see the brain tissue looks kind of homogeneous, although it's flecky. After, you got these big holes. Not unlike what you might see with Alzheimer's, right? You get these plaques and the brain stops working where there's a big hole, right? This is a problem, obviously. There are a lot of different animals that have this problem. We give it different names. So scrapie or scrappy, I'm not sure how to pronounce that, in sheep. They used to grind up the sheep, put it back into the feed, and then feed the sheep. Guess what? If the disease was in the sheep food, the disease became part of the sheep in the next round. Kuru. Now they discovered this one down in Papua New Guinea, where different villages were always warring with each other, kind of limited resources issues. And if you beat another village, it was sort of part of the tradition was to eat the brain of your defeated villager. So they're transferring it that way to each other. And in the 50s, this was discovered, and it took a while, but they eventually convinced them to stop doing this. And then the disease continued to show up because it's got a long latency period, but I think it's largely been removed from those populations now. Creutzfeldt-Jakob, this was one that was found in, I believe, Europe, and those are I think Creutzfeldt was the doctor, Jacob may have been the individual. Same sort of manifestation, all of these. Diseased brain tissue, holes, and lack of coordination and eventually death. Bovine spongiform encephalopathy, BSE. When I was doing this in the early 2000s, this was a big story, and I was a little bit concerned because it was big in Europe. They were afraid that it was becoming a problem. It was, and I was eating some cattle tissue in Austria, and I was kind of wondering, what the heck? Is there any brain tissue in what I'm eating here? I don't think so. It was just a fancy form of a Wiener Schnitzel. It's kind of hammered out meat and then deep fried. But some of that meat was coming from the face of the cattle, and I thought that's too close to the brain. So I was worried. And then, of course, on our deer and our elk and such is the CWD. So what is this? It's not a microbe. Okay, these are not microbial diseases. These are caused by proteins, as you probably know by now. They're called prions, and our brains have prions, and I'm not sure we have a full handle on what they do, but they're in there. They do things. And the left-hand side is a typical prion. And just so this is a protein, it just shows you, so this is probably just the amino acid stretch over here, okay? And so it just has these amino acids. When you get to a certain way in terms of how the amino acids are lined up and which amino acids, they go into something a lot like the double helix in DNA. They form an alpha helix, single-stranded helix, okay? And oftentimes that's an important part of a protein, so it's involved with whatever enzymatic activity it does. You want your particular, I'm just making this up here, but you want your particular area to be exposed for the enzymatic activity, and it's being positioned by the presence of these alpha helices, okay? If you really have an important factor and you need that position exactly folding exactly right so it's in the right position, then the proteins go through what's called beta sheets. I'm not going to go into how that happens, but it's a much more stable version than the alpha helix, okay? And look what happens at the conversion, right? It goes, so an analogous situation before I move on, the beta sheets might be considered similar to a covalent bond, whereas the alpha helix is hydrogen bonds. And if you heat up the alpha helix, the hydrogen bonds relax and the system kind of falls apart. But that doesn't happen with these beta sheets or a covalent bond. They're stuck. okay, in that position, in that orientation. So look what happens when the bad protein comes in contact with a good protein or a normal protein. It converts it and creates this huge beta sheet. And now it's a problem. Okay? I don't know what it does for the functionality, but what it does definitely do is create a very stable protein that isn't going anywhere, is going to convert others into that same morphology and cause plaques and problems in our brain tissue. So the abnormal prions are replicating by converting normal proteins. They're not really replicating the way we do with our DNA. They're just converting good proteins into bad proteins, which convert more good proteins into more bad proteins, etc. The other factor is those beta sheets make them extremely stable. So you can take all your animals and burn them, and you're basically, it's like the old plague storyline where, let's see, ashes to ashes, we all fall down. The ashes coming down from the burning of the bodies was spreading the disease, okay? And that's probably what's happening with these proteins when they burn the bodies. It's spreading, the ashes are spreading the disease beyond where you're burning. And they don't break apart with high temperature. In fact, they get into the soil. And they've gone back into some places and dug up the soil, and they're still there years later. And I talked to one woman at Upstate. She actually came in and gave a presentation at our class. She said that one of the things they do, if they've had medical equipment that they've used to dissect brains or something that's been exposed to these prions, they autoclave them for, I forget, but it's a crazy, like an hour at high temperature in super caustic, I believe acid, but it may have been basic solutions. And she said, and sometimes that's still not enough. The proteins are still there doing their thing, whatever their thing is, not doing their thing, but basically capable of converting good prions to bad prions. So high pressure, high temperature in the autoclave in a very caustic environment, and they're still not disassociating. So that's why it's a problem. To put it into sort of a closer to home reality check, there are a number of states where it has been seen in native populations of our hoofed animals, but in New York it was found up near Rome a few years ago. That was probably I think that was a game farm. I don't know if it's been found in the native populations, but in the game farm there's lots of close contact. Remember, this is how diseases spread. Our old logistic curve and density dependent issues where you have too many bodies, not enough food, and too much disease spread because they're all in close contact. So they're in a game farm, the animals are in close contact, and that's how it spread. I think they kept it in check, but they did come up with some very specific recommendations. So here's where Rome is, right in here, and so this was a big area where I think hunting was off limits so people wouldn't get hurt. This is a number of years ago, and sorry I didn't keep up and follow up on that, but maybe we'll do that in our discussion groups. And then this is something out of the CDC, basically just saying don't eat brain, eyes, spinal cord, tonsils, intestinal tract, spleen, or retropharyngeal lymph nodes. Deal. I'm sold. I don't think I would anyway, but maybe some people prefer these things, like the flavor. They're sort of the sweetbreads concept. Don't do that with your hunted animals. And the two genera that they're highlighting here are Cervus and Odocoileus. I don't know how to pronounce that one, but I believe that's the deer and the elk. So, right. Lastly here with the COVID storyline, one more time. I just pulled this off the web on July 14th from the CDC, and it's showing you the number of, let's see, are these deaths? Oh my gosh, I forgot. I believe these are infections. No, infections are in the hundreds of thousands. Worldwide deaths, these may be deaths. I'll come back and let you know. But the general trend, I think, is the interesting thing. What we're seeing is we flattened the curve in New York. It was horrible. Again, look at the y-axis, 20 to 40,000, okay? And the different lines are basically individual models, and then the single line, the black lines, are sort of the putting all those different models into one and coming up with a consensus pattern. Nice and flat. We're not really increasing COVID as of July 14th anymore. But look at Montana and any of these other states that you may be hearing about or have heard about that managed to avoid the problem when New York was getting hammered in the spring in March, April, May, June. Montana wasn't having very many infections. But now look what's happening. Now it's happening. And again, the y-axis is very different. We're in the hundreds rather than the tens of thousands. But the curve is such that those numbers are doubling on a regular basis. We will be in the thousands and tens of thousands, I suspect, if there's enough people in Montana very soon. This is a problem for Montanans. Could ignore it. They could go to their bars. They could leave their masks off. They can't anymore. Okay, this is really a problem. So, as I said, the New York curve has flattened, whereas Montana is sweeping up. And do pay attention to those y-axes when you look at graphs so that you can see that, wow, this looks tremendously troublesome, and it is, but the numbers are nowhere near what New York had just because of populations of Montana versus New York City. Okay? And remember that we have these SIR models, and if we can come up with a vaccine, I've kind of created a new category where we could have the recovered individuals basically have, through the vaccine, it replicates moving from infected to recovered, okay, and getting out of the susceptible with the vaccine. And so please do use vaccines. Okay. So to summarize this chapter, parasites and pathogens have important ecological impacts on host species. And in some cases, you can have outbreaks. Think of the chestnut blight. It could be beyond an outbreak. It could be devastating for the host until someone comes along and comes up with a way to generate resistant varieties. But we can also pay attention to, if it's human, which is usually one of our more important host species in our minds, we can use vaccines to help avoid some of the more intense impacts or alleviate them. Parasites and pathogens and their interactions are impacted by resistance and defenses, virulence and offensive strategies. So these are where we can play around with our models and come up with some interesting predictions and as well as management strategies to deal with the problem. Remember that transmission can be horizontal or vertical. At the same time, remember that resistance can be horizontal and vertical. Go back to the unit that I talked about resistance to pull that up. And that SIR model is important, and we can use it for the human situation with COVID right now. I suspect there's going to be a question like that on some future exam in my class, applying SIR to COVID. I have no idea what the question is yet, but it's starting to gel in my brain. So yeah, you might as well get that one down now and get it internalized so you can think about it on a test and come up with the right answer. So that's it for today. That's today's segment. Next chapter is going to be fun for me. It's on mutualisms. The book finally included mutualisms. There have been chapters in the past on mutualisms, and they completely just basically said, yeah, they're there, and moved on and talked about anything but mutualisms in the chapter. This particular book does a little bit better, but they still are missing some really key interactions that I want to have you guys understand. And in the end, I'm going to go, of course, into my research a little bit, or at least into the topic of my research, and that is mycorrhizal fungi and plants. So classic mutualism that is basically ubiquitous all over the planet. Doesn't matter ecologically. Let me restate that. Doesn't matter ecologically. It's a mutualism that is ubiquitous all over the planet. It must be it's important. So let's go there, next chapter. Okay, so that's it for this segment. We'll see you next time. Cheers.